Gender-specific offspring. A new method tested on mice is 100% effective. Using gene editing, scientists have successfully bred litters of mice with a specific sex, either exclusively female or exclusively male. The technology, developed by experts from the Francis Crick Institute and the University of Kent, has demonstrated 100% success. In scientific research, as well as in agriculture, there is often a need to have animals of one specific sex. For example, in experiments on male or female reproduction, only individuals of the sex being studied are needed. In agriculture, on the other hand, only female animals are needed for egg and milk production. This situation leads to the common practice of removing animals of the unwanted sex after birth. The new method uses CRISPR-Cas9 gene editing technology and is 100% effective. The study was published in the journal Nature Communications. Sex selection relies on the CRISPR-Cas9 gene editing method, which allows for the removal of an unwanted DNA fragment from the gene being studied and the insertion of a different sequence in its place. The key here is the Cas9 enzyme, which cuts DNA, allowing scientists to modify the genome. Guide RNA, gRNA, is also crucial, as it indicates the cleavage site to Cas9. The team placed the enzyme and the gRNA separately in the parents' bodies. One of the elements was located on the father's X or Y chromosome, meaning it would be inherited only by female or male embryos, respectively. The other element was implanted in the mother's body and is inherited by all embryos. Thanks to this forced mutation, an embryo with the unwanted sex is unable to develop beyond a very early stage of approximately 16 to 32 cells. Using this method, researchers were able to control the sex of the litter with 100% success. To create an all-male litter, the researchers edited the father's X chromosome, meaning only females inherited the mutation, and to create an all-female litter, they edited the Y chromosome. Interestingly, this method did not lead to a drastic reduction in offspring, with litter sizes ranging from 61 to 72% of control litter sizes. The researchers suggest this is because animals like mice produce more eggs than required during each cycle, allowing some to be lost during early development without reducing litter size. According to the researchers, the new method will enable birth control in other mammals as well. Experts also emphasize that gene editing has no harmful effects on surviving offspring. Our work could have an immediate and valuable impact on research laboratories because we've shown it to be safe and effective in mice. Many studies require both sexes, but there are areas of science where only one is needed, says co-author James Turner from the Sex Chromosome Biology Laboratory at the Crick. The implications of this work are far-reaching in terms of improving animal welfare, but they should also be considered ethically, says study co-author Peter Ellis from the University of Kent. In particular, before any agricultural use, extensive discussions and debates should take place. Legal changes may also be necessary, he adds. Strength training good for Alzheimer's disease. Regular physical exercise, particularly strength training, can delay the onset of Alzheimer's symptoms and even prevent the disease, according to research by Brazilian scientists. This type of exercise, the researchers emphasize, is a simple and inexpensive therapy for Alzheimer's patients. According to a paper published in Frontiers in Neuroscience by researchers affiliated with the Federal University of São Paulo, UNIFESP, and the University of São Paulo, USP, strength training can offer significant benefits to those with Alzheimer's disease. Research suggests that engaging in physical activities that improve strength and endurance, such as weightlifting, may delay or even prevent Alzheimer's disease. A method highlighted in recent studies is strength training, also known as resistance training. The World Health Organization generally recommends this type of exercise because it contributes to improved balance, body development, and helps prevent falls. Engaging in this type of physical activity is crucial for building muscle mass and increasing muscle strength, while also strengthening bones, which then increase bone density. This is also important for our health, as it can help prevent sarcopenia, a condition in which muscle atrophy occurs. At the same time, there is increasing awareness that this type of physical activity can also have a significant impact on the physical and mental well-being of people with Alzheimer's disease. However, as is well known, engaging in such strenuous exercise for older people or those already suffering from dementia can pose significant challenges. However, resistance training is increasingly proving to be an effective strategy for managing Alzheimer's disease, which has a multifactorial basis, but not, for example, one caused by a specific inherited genetic mutation. However, it can delay the onset of the disease in people with a family history of the disease. To determine how strength training can slow the progression of Alzheimer's disease, scientists decided to conduct research on transgenic mice. Their genotype was modified with a mutation responsible for the deposition of beta-amyloid protein in the brain, forming so-called senile plaques. As these plaques accumulate in the central nervous system, they impair synaptic connections and damage neurons, leading to the development and progression of Alzheimer's disease. These mice underwent a fairly intense training regimen. They were trained to climb a 110 cm long ladder with an 80-degree incline, with rungs spaced 2 centimeters apart. Additionally, weights weighing 75%, 90%, and 100% of their body weight were attached to their tails, mimicking certain types of resistance training seen in gyms and fitness centers. The entire exercise cycle lasted a total of 4 weeks. After completing the exercise, blood samples were collected from the mice to measure their plasma corticosterone levels. Corticosterone is the equivalent of cortisol in humans, and elevated levels in response to stress increase the risk of developing Alzheimer's disease. However, in the trained mice with the genetic mutation, corticosterone levels were within the normal range, similar to those found in mice without the mutation. The studies described above indicate that intense physical exercise can not only inhibit but even reverse the adverse neuropathological changes associated with Alzheimer's disease.